Hi, my name is Douwe CNS and I'm part of the Cognitive Neurophysiology Lab at Albert Einstein College of Medicine. Today, I will be presenting data that is part of a larger project on 22q11.2 deletion syndrome. In this project, we are characterizing cognitive and brain function in 22q11.2 deletion syndrome. This syndrome is characterized by developmental delays, cognitive deficits, and neuropsychiatric disorders. Here, we were particularly interested in the high incidence of schizophrenia in this population. In this study, we investigate basic auditory processing and sensory memory using EEG in a group of adolescents and adults with 22q, with and without psychotic symptoms. We chose these processes because we know that those are two of the processes consistently reported as impaired in schizophrenia. We tested 26 neurotypical controls and the same number of individuals with 22q. We divided the 22q group in two. 22q minus group, 11 individuals with 22q but no psychotic symptoms, and the 22q plus group, 15 individuals diagnosed with 22q that have one or more psychotic symptoms. We collected EEG data while the participants watched a movie without sound while listening to a passive audible paradigm. The standard tone, presented 85% of the time, was 100 milliseconds in duration. The deviant one, of 180 milliseconds in duration, was presented 15% of the time. These tones were presented in several randomized blocks with different SOAs, or presentation rates, that go from the start of one tone to the start of the next one. We used a 450, 900 and 1800 millisecond SOA. We did this to impose different demands on sensory memory. Shorter SOAs impose less of a demand on sensory memory since it's easier to retain the previous tone when the next one happens within a shorter time frame. We also assessed the participants' IQ and working memory and investigated the presence of psychotic symptoms using the SCID. Here, when we look at the IQ, we see in blue the neurotypical group, in green the 22q minus group, and in purple the 22q plus group. Both 22q groups showed on average an IQ of around 70, which is similar to what is reported in the literature and significantly different from our neurotypical controls. We see a similar pattern when we look at the working memory. When we move over to our EEG data, we're looking at the N1. Only standards are plotted here, and it indexes basic auditory processing. Again, we have in blue the neurotypical group, in green the 22q minus group, so the 22q group without symptoms, and in purple the 22q plus group, or the 22q group with symptoms. We see per group three waves per group, which represent the different SOAs. As is expected, the shorter SOA, this one, the smaller the amplitude, which reflect neural adaptation. As we can see here, adaptation and basic auditory processing seems to be intact in all groups. Interestingly though, whereas the 22 minus group presents increased amplitudes when compared to the neurotypical group, the 22q plus group presents decreased effects of adaptation compared to the 22q minus group. Similar reductions are consistently described in schizophrenia, and importantly, we have observed this same pattern of increased amplitudes in those without psychotic symptoms and decreased amplitudes in those with symptoms in other tasks, for example visual tasks, that we have done in individuals with 22q. Here is the MMN. The MMN indexes sensory memory. It is presented as the difference between deviance and standards. The difference shows if the brain is able to tell the difference between two sounds. Here, the MMN seems to be generally intact for all groups, but we see an increased MMN for larger SOAs for both 22q groups regardless of the presence of psychotic symptoms. In conclusion, our findings seem to suggest the presence of two distinct mechanisms. One intrinsic to 22q resulting in increased N1 and MMN responses, another related to psychosis leading to a decreased N1 response. Thank you for your attention.